Stanford University.
So my talk will of necessity be an overview because we could have an entire course just on targeted therapies, as I hope you're aware. I just want to go back and reflect on a very famous paper that you've all read from Hanahan and Weinberg. This was a review and a thought piece in Cell in the year 2000, most of which is still relevant today. The paper is called The Hallmarks of Cancer. And basically, Hanahan and Weinberg divided the properties of cancers into 6 general areas that might be therapeutically exploitable. These would be self-sufficiency and growth signals, and I should say that many of these properties also involve critical pathways for oncogenesis. Tumor suppressor genes and oncogenes will affect some of these properties. Self-sufficiency and growth signals is one of those. Insensitivity to anti-growth signals, tissue invasion and metastasis, a limitless replication potential, so the role of telomeres and senescence seems to be lost. Sustained angiogenesis, which has become an important target in chemotherapy, and then evasion of apoptosis, so changes in how cells respond to injury and whether they go on to die or to survive. In this paper, they draw attention to our knowledge of the increasing complexity of the regulation of various cell processes, including growth and survival. And they talk about the integrated circuit of the cell. This is obviously important when we disrupt cell pathways with targeted therapies. And an abiding challenge is to understand what the key regulators of survival and growth in individual cancers are, and how we can most efficiently combine inhibitors of those functions in the cancer cells specifically. So this field was notably advanced by the Human Genome Sciences, the Human Genome Project, which was completed with the complete sequencing of the human genome by an NIH consortium and by Celera about 5 years ago. For targeted therapies, it's important to note that in the human genome, there are 518 known protein kinases. These are the on-off switches for many, many different cellular processes. 90 of these are tyrosine kinases, 428 are serine threonine kinases. There are both monoclonal antibody drugs against receptor kinases on the cell surface and small molecules that inhibit usually the kinase function of these enzymes, whether they're cytosolic or transmembrane proteins. And so most of the new targeted drugs are in fact targeting the human kinome, the kinase family. So currently, there are more than 600 kinase inhibitors. They're in various stages of preclinical and clinical development by more than 200 companies. And noted here is the fact that around 50 angiogenesis inhibitors are already in clinical trials, 26 VEGF receptor inhibitors. This is a very crowded field. And one of the interesting challenges in cancer drug development is how to most efficiently and accurately assess the optimal doses and schedules and side effects and relative efficacy of these drugs. That's an enormous challenge. So within these current and